السلام عليكم ورحمة الله وبركاته. My lecture today in medical mycology. Specifically, it's a general introduction on fungi. Fungi are eukaryotic microorganisms that occur in two basic forms, as you see in this picture. These forms are yeast or molds. Yeast are single-celled forms that reproduce by budding. Molds is a fungus that grows in the form of multicellular filaments called hyphae. Hyphae can be sparsely septate to regularly septate and possess a variable number of nuclei. So what is hyphae? Hyphae are the long filamentous branches found in fungi and Acinetobacterium, as shown below. Hyphae are important structure required for growth in these species, and together are referred to as mycelium. Some fungi are dimorphic, which means occurs as a combination of both forms. So, the definition of dimorphic fungi is a fungi growing as yeast or spherules in vivo, as well as in vitro at 37 centigrade, but as molds at 25 centigrade. Now, I was talking about fungi reproduction or multiplication. Fungi reproduce sexually and or asexually. Perfect fungi reproduce both sexually and asexually, while imperfect fungi reproduce only asexually by mitosis. Fungi reproduce asexually by fragmentation, budding, or producing spores, the most common mode of asexual reproduction. Spores are microscopic biological particles that allow fungi to reproduce, serving a similar purpose to that of seeds in the plant world. Spores may be either sexual or asexual in origin. In both sexual and asexual reproduction, fungi produce spores that disperse from the parent organisms by either floating on the wind or hatching a ride on an animal. Note: spores form only in molds forms. In general, sexual reproduction occurs by the fusion of two haploid nuclei, karyogamy, as you see in this diagram, then followed by meiotic division of the diploid nucleus, forming sexual spores, while asexual reproduction, as you see, result from division of nuclei by mitosis, forming gonidia, which is asexual spores. There are different types of sexual spores as well as asexual spores, gonidia. So as you see in this diagram, sexual spores are an oospore, which is a thick-walled sexual spore that develops from a fertilized oosphere in some algae, fungi, and oomycetes. Second, zygospores. Zygospores are single large spores with thick wall. Third type of sexual spores is ascospores, are spores formed in a sac called ascus. Fourth, basidiospores are spores formed external on the tip of pedestal called basidium. Note, imperfect fungi are fungi that do not form sexual spores or asexual spores. Let's move to talking about types of asexual spores. Gonidia. Gonidia are asexual spores formed from the side or end of specialized structure. Their properties like color, shape, and arrangement are important in the identification of fungi. There are different types of gonidia, which is asexual spores. Types of gonidia or asexual spores are, as you see, arthrospores, chlamydiospores, blastospores, sporangiospores. Arthrospores are a very primitive spore type formed by the breaking up or dysregulation of fungal mycelium. Second, chlamydiospores formed within hyphae, thick-walled spore, whereas blastospores, a bud coming off the parent cell. Sporangiospores is 100 of asexual spores formed within a sac called sporangium at the end of the aerial hyphae. Now, I'll move to talk about fungi structure and start with cell wall structure. As it appears, and as you see in this picture, in contrast to bacterial cell wall that consists of peptidoglycan, the rigid cell wall of fungi is a stratified structure, consists of chitinous microfibrils embedded in a matrix of small polysaccharide, proteins, lipids, inorganic salts, and pigments that provides skeletal support and shape to the enclosed protoplast. Therefore, The antibiotics that inhibit peptidoglycan synthesis, the component of bacterial cell wall, such as penicillin, cephalosporin, and vancomycin, are not effective against fungi. Now I'll move to talk about cell membrane structure. The function of a plasma membrane is regulation the passage of materials into and out of the cell by being selectively permeable. Membrane sterol provides structure, modulation of membrane fluidity, and possibly control of some physiologic events. The question is, why antifungal antibiotic such as the azole drugs and amphotericin B are not affect bacteria or mammalian cells? As you see in this picture, in contrast to the mammalian and bacterial plasma membranes that contains cholesterol and peptidoglycan respectively, fungal cell membrane contains ergosterol. So several antifungal agents interfere with ergosterol synthesis. Therefore, antibiotics that inhibit ergosterol synthesis, such as the azole drug and amphotericin B, are not affect bacterial or mammalian cells. But what is the mechanism of antifungal that inhibit ergosterol? The mechanisms that inhibition of ergosterol synthesis by antifungal agents can result in general from the activation of chitin synthase, zymogen, leading to excessive chitin production, causing abnormal growth. Let's move to next slide to talk about the virulence factor or the pathogenic factor. Pathogenic or virulence factor. There are different virulence factors in fungi. First, the ability to grow at 37 centigrade, which is one of the most important pathogenesis factor in fungi. Second, keratinase production. Production of keratinase allows dermatophytes to digest keratin in skin, hair, and nails. Third, dimorphism phenomena. Dimorphism allows many fungi that exist in nature as molds to change to a yeast form in the host and thus become pathogenic. In contrast, Candida albicans exist in the yeast forms as normal flora, but becomes invasive in the filamentous form. virulence factor is capsule and the adherence ability. The antiphytocytic properties of the Cryptococcus neoformans capsule and the adherence abilities of Candida albicans allow pathogenic potential for these fungi. These fungi are phagocytosed by alveolar macrophages, but are not destroyed. Instead, the fungi are spread hematogenously to distant sites in the body. There is one exception, which is Cryptococcus neoformans, which is disseminate without being phagocytosed. Fifth, virulence factor, the ability to causing allergic reaction. The pathogenesis of some fungi may be at least partly due to the host's reaction to the organism, such as the allergic reaction elicited by some fungi. Let's move to next slide. So, this slide illustrates different diseases that caused by fungi. Mycosis. So what is mycosis? The diseases caused by fungi called mycosis and can be divided into 4 categories. These are cutaneous mycosis, also called dermatophytosis. We will take an example about this type of disease, tinea, or called ringworm. Second mycosis is subcutaneous mycosis, and example sporotrichosis. Third, systemic mycosis, example histoplasmosis and blastomycosis. The last one, opportunistic mycosis, and we will talk about aspergillosis and candidiasis as an example. In the next lecture, we will talking about all of these diseases separately. In conclusion, fungi are eukaryotic microorganisms. Can be unicellular, multicellular, or dimorphic, which is when the fungi is unicellular or multicellular depending on environmental condition. Third, fungal cell walls are rigid and contain complex polysaccharides called chitin at structural strength and glucans. 4. Ergosterol replaces the cholesterol found in animal cell membrane or mammalian cell membrane. Fungi reproduce sexually and or asexually. Finally, fungi can cause different types of disease, such as cutaneous mycosis, dermatophytosis. and subcutaneous mycosis, systemic mycosis, and opportunistic mycosis. Thank you for your listening. Please don't hesitate to ask me any questions.